Morning.
Good morning.
Morning. Internet issues. Back on. Okay, yeah, just log back in if you get booted out. Not a problem. All right. Well, let's go ahead and get going here. I think I've got everybody checked in. It looks like one or two of you might be having some internet issues, so no worries. All right. So a little bit of stuff I want to cover real quick before we move on to the meat of the presentation, or the meat of the information that I want to get through today. I want to just talk about a couple of additional things that maybe would have fit in better yesterday, but the sake of preventing saturation of topics, we blessedly decided to call it yesterday. So a couple of additional things I want to talk about, just because they are becoming quite severe issues. And that is the concept of antibiotic resistance. It's becoming a real big issue. It has over the last couple of decades, but so for the last couple of decades, many of the bacterial infections that we would be able to easily treat have become much harder to treat, and that is because of those bacteria are becoming and have become quite resistant to many, and in some cases all, of the antibiotic options that we have available. And this is in part due to the fact that, as we talked about yesterday, bacteria, the way they're able to divide, the way they're able to share genetic material through like plasmid conjugation and other mechanisms, they're very easily able to acquire traits that allow them to overcome the antibiotic, because antibiotics work molecularly. They work by, for example, the cell wall inhibitors like the penicillins work on degrading a chemical structure in the walls of particularly gram-positive bacteria called called the beta-lactam ring, and they degrade that ring, and then that kind of opens the bacteria up, and it ends up dying. So they are toxic, bactericidal, as we say, in many cases. Other antibiotics work by inhibiting the ability for the bacteria to produce protein. Bacteria have ribosomes, just like we have ribosomes in our cells, although the subunits of the ribosomes are different in animal and plant and animal and bacterial cells. And so we can make antibiotics that target those ribosomes and prevent them from creating proteins, and then they can either quit working, and then our immune system takes care of it. And whenever an antibiotic prevents a bacteria from growing and kind of doing its job but doesn't directly kill it, we call that a bacteriostatic antibiotic. Something like azithromycin, or a macrolide like azithromycin, is at some certain doses at least bacteriostatic. But then again, the bacteria can, through acquiring mutations, right? And this really is just evolutionary theory. This is just natural selection. Essentially, what's going on is you introduce a selection pressure, i.e. an antibiotic, and some number of bacteria are going to have genes just through random mutations, because they're constantly dividing, right? Constantly dividing, and a generation for a bacteria can be minutes versus 20 or so years in a human. They're able to divide very rapidly, and if one bacteria has a gene that makes it a little more resistant, say, to an antibiotic, and it survives that onslaught of the antibiotic, then that bacteria might be able to survive, and then it can divide and it can propagate that gene, and then the next generations are a little more resistant to that bacteria, and then you acquire random mutations that can make them more resistant, and the bacteria that acquire mutations that don't help them end up dying off, and over time you end up developing highly resistant bacteria that are highly resistant to that antibiotic. And that's kind of what we're seeing play over and over again. And there are certain infections we do not have we do not have the capability of treating certain infections. For example, gonorrhea. There are now types of gonorrhea out that are completely untreatable. That is to say, we have no antibiotics whatsoever that can treat them. There are types of tuberculosis now, something called XDR-TB, or extreme drug-resistant tuberculosis. No way of treating it. And we see in some areas of the world going back to pre-antibiotic times where we were taking people and essentially locking them away in asylums and kind of like, well, hopefully your immune system can do something about this because we don't have anything for you. In other cases, such as Clostridium difficile or C. diff infections, which infects the gut and causes a severe diarrhea, can be life-threatening. In some cases, we have to resort to very novel kinds of measures to try to treat those infections. In fact, the standard of care for C. diff infection, because C. diff is highly antibiotic resistant and it tends to occur in people that have taken antibiotics. It's something referred to as a super infection. Anybody who goes on an antibiotic is actually prone to developing super infections because the antibiotics alter the microbiome of their mucous membranes of their gut primarily. And you take out a bunch of bacteria in the gut, and then other bacteria that are drug resistant can come in, or maybe they were always there, but they were kept in check by the other organisms in your gut. They're no longer kept in check and they start growing. And now you have this infection that you can't treat. But the standard of care for treating something like C. diff is actually something called a fecal transplant. And a fecal transplant is essentially where you obtain fecal material from a healthy person and put that fecal material into the person with C. diff with the hopes that that normal microbiome that you took from somebody else can repopulate the gut of that sick person and take over and hopefully treat that infection. So this is becoming a big problem, and the overuse and misuse of antibiotics is accelerating this. This has accelerated this. There are a lot of people are put on antibiotics that don't need to be on antibiotics, particularly people that have upper respiratory infections, viral infections. Antimicrobial therapy does not work against viruses. So about 80 to 90% of the time, if you get a cold, you get a scratchy throat, and you cough and you sneeze and you don't feel great, about 90% of the time, that's a viral infection, right? That's one of the many coronaviruses. It's a rhinovirus. It's a parainfluenza virus, right? It's a virus. So putting somebody on antibiotics is not only ineffective, but it's actually harmful. And unfortunately, we're seeing a lot of this happening right now, where people, for reasons that I don't understand very well, because there isn't great evidence for this, but people are being placed on zithromycin, Z-Paks, that get COVID. So again, if someone has a viral infection, you're just throwing antibiotics at them, and this is adding to the problem of antibiotic resistance. And to make matters worse, it's been a couple of decades, actually over a couple of decades, since we've developed any new antibiotics. And there is not a whole lot of incentive for drug companies to develop new antibiotics. It takes some hundreds of millions of dollars to develop a new drug, get it to the market, and then within a few years, you already start seeing resistance. And people don't take antibiotics for an extended period of time, right? You get an infection, you go on an antibiotic, hopefully the infection clears, and then you don't take the antibiotic anymore. And so it's very hard in some sense for drug companies to recuperate the cost of developing these antimicrobial therapies. It's far more better for these companies, at least from a financial standpoint, to develop medications that people take for multiple years, if not for their duration of their life. Right? That's really where a lot of the money is, the financial incentive. So you look at the top selling medications, it's not antibiotics, it's not immunizations or any of that. It's drugs that people take on a daily basis, day after day, year after year, decade after decade. So you're looking at medications to treat hypertension, antihypertensive agents. You're looking at agents to treat diabetes, right? Type 2 diabetes specifically. You're looking at antidepressants. You're looking at anti-anxiety medications, things like that. Those kinds of medications are really where the money is at and where the incentive in drug development comes from, because we know people are going to be on those medications for extended periods of time. So antibiotic resistance, huge problem, and it's only getting worse and worse. And we are running out of lots of options. If not, we have no options to treat certain infections. It's just, and it's honestly, it's absolutely terrifying to think about something like gonorrhea being untreatable, because gonorrhea, when I was coming up, was an afterthought. If someone got gonorrhea, it's like, oh well, you dummy. All right, let's get you covered. And in a week they're fine. And now there are people, it's kind of like, well, I hope you don't get pelvic inflammatory disease and either die or, you're sterile for the rest of your life. I hope that doesn't happen, because we don't have great options to treat you. So yeah, there's some really scary stuff happening there. And this is not only does it impact us, but it will continue to impact us in the pre-hospital environment, because we end up treating and transporting a lot of these people with these drug-resistant infections. So I wanted to just mention that. Okay. So some other things I want to talk about. I want to spend a little bit of time talking about cancer. We mentioned it a little bit yesterday, but I just wanted to fill in some additional pathophysiological gaps, if you will, with cancer, because cancer is very common, right? It's an incredibly common cause of morbidity and mortality. About 50% of us will come into contact with cancer in our lifetimes. It's in the top 3-4 causes of death nationwide. So it's relevant, and it's something that I want to just talk about a little bit. And cancer is just not one thing. Cancer is a large, diverse group of disorders, and we talked about kind of what the pathophysiological character of cancer is. These are cells, these are abnormal cells that are dividing unchecked, and they're taking resources from the body, and they're causing organ dysfunction, right? Because they're not doing their job. They've lost the ability to do their job. They're not responding to the immune system. They're dividing uncontrollably, and can, in many cases, cause problems. And interestingly enough, we all get cancer. In fact, we get cancer multiple times throughout our lives. It's just that in most cases, our immune system is able to identify those abnormal cells that are growing, right? Identify them and able to target them and cause them to undergo apoptosis and prevent that cancer from becoming a substantial problem. It's just that in certain cases, as we talked about, those cancers, those cells have genes that code for different proteins, like major histocompatibility complex proteins, that prevent them from responding to the immune system, right? And so the immune system can't tell those cancer cells to go away, to die. And so that allows those cancer cells to evade, to escape the immune system, if you will. And so what do? Well, we tend to give drugs. So there are really 3 major ways we treat cancers. You can treat them with drugs called chemotherapy. Chemotherapy is probably not a great term. I think in the vernacular, we all understand what it means, but chemo literally means chemical. It's chemical therapy. And so an antibiotic for bacterial pneumonia is chemotherapy for an infection, but there's chemotherapy for cancer as well. Interestingly enough, many of the cancer drugs that we use are old antibiotics that were too toxic to be used therapeutically. And in many cases, what these agents do is they induce mutations they cause mutations that are so bad that it prevents cells from dividing. In some cases, they target and they prevent the cells from dividing, but in a lot of cases, they induce fatal mutations. A lot of them are what we call alkalizing agents, and they induce a certain kind of mutation, and so they essentially prevent cell division, or when cells are dividing, that's when the DNA, because typically when we talk about the DNA and the chromosomes, which we'll mention here in a little bit, you don't literally see the chromosomes all lined up in the cell or in the nucleus, right? There are only very special times when the chromosomes are lined up and just out in the open, so to speak, within the cell during cell division. We'll talk about that in a little bit. But it is those chromosomes and the genes are like wrapped up and protected, like wrapped around big old histone proteins and compacted and protected inside of the nucleus. And then there are special proteins that come and kind of open them up and unzip them, DNA, helicase, and things like that. That, if you need to code for a specific protein, they find that specific gene, unwrap the DNA from the histone, copy it into RNA, and then it gets wrapped back up and protected. But cells that are rapidly dividing, right, their DNA tends to be open more, right, because they're copying DNA more. And so rapidly dividing cells are more prone to developing mutations and problems that can lead to cancer. And so it should come as no surprise that the more common cancers that we see are in tissues where you tend to have a lot of cell division, right? For example, skin, right? Cancers involving the skin are very common, whereas cancers that involve areas where you don't have a lot of cell division, such as the brain and the heart, are fairly rare. They still exist, but they're relatively rare compared to, say, cancers of the skin, cancers of the gut, the gastrointestinal tract, cancers of the respiratory tract, cancers of the blood. You have an incredible amount of cell division. Those stem cells, as we talked about yesterday, are pumping out white and red blood cells all day long. And so it should come as no surprise that, leukemias, lymphomas are relatively common as far as cancers go. All right. So when we talk about cancer in general, the main risks really come down to a combination of environmental factors and genetic factors. And it's kind of a complex interplay between the two. So we talk about environmental factors, exposure to situations that will result in causing mutations or activating and deactivating certain genes, and we'll talk about some of those genes here in just a bit, are what we're concerned about. So exposure to ionizing radiation, for example. Not all radiation, right? But what we call ionizing radiation. This is radiation that has a sufficient amount of energy to knock electrons off of atoms. And when you do that in the setting of a chemical bond, you break that chemical bond, right? You ionize it. And so you're talking about high energy radiation. So ultraviolet radiation, right? For example, UVA, UVB exposure in the skin can increase the risk of certain cancers. You're talking about X-rays, gamma rays, alpha particles, beta particles, neutrons, and those kinds of things, which have a lot of energy, right? So ionizing radiation, different pollutants and toxins, right? Exposure to smoke, cigarette smoke, for example, is one of the biggest risk factors for cancers of the respiratory tract and other areas of the body, for example, right? So ionizing radiation, toxins, right? Sometimes you can have injury and constant inflammation. For example, people have reflux disease. I think I mentioned this earlier. They get reflux in their esophagus, and over time, that constant exposure to stomach acid kind of erodes the esophagus, and those cells of the esophagus undergo changes, and it turns into what's known as a Barrett's esophagus. And then a large percentage of Barrett's esophagus patients will go on to develop esophageal cancer. In fact, that's what happened to my grandfather. Had Barrett's esophagus and ended up developing esophageal cancer and subsequently died of esophageal cancer. So you can see there are many different environmental factors, and then there are genetic factors as well. There's certain genes, and there's this concept of the genome. I think I mentioned this yesterday, right? Or the genotype. Those are your genes. And then there is something called the phenotype. And the phenotype is what genes are actually being expressed, right? Because not all genes and all cells are turned on all the time, right? For certain cells to specialize, many of those genes need to be turned off. They need to be suppressed. And then other genes will be expressed and the cell will specialize, right? And that's called the phenotype. So you've got your genotype and your phenotype. And there are certain genes, and many of these genes are associated with growth and development, but they are known as proto-oncogenes. And by themselves, these proto-oncogenes generally aren't a problem, but what can happen is some toxin can come along, ionizing radiation, some chemical injury can come along and can cause some mutations or some changes and can flip those proto-oncogenes on. And when that happens, or in some cases, you can just have a abnormally functioning gene, and this is just referred to as an oncogene. And many of these oncogenes code for growth. And so you can see that if you have an oncogene that codes for very aggressive cell growth and division, if that gene is not turned off or suppressed, it can lead to cancer, right? Or it can be a big part of cancer. And then on the other hand, there are what we call tumor suppressor genes. And these are genes that tend to suppress oncogenes, right? And essentially keep them, suppress them, like, okay, we had some growth during childhood, and now we need to turn that growth off or suppress it. And that's what the tumor suppressor genes do. So you have some genes that suppress other genes, right? So it's a very complex interplay of things. And these tumor suppressor genes are very important because if you have abnormal tumor suppressor genes, that can also play a big part of cancer. In fact, these are so important. I'm just going to give you examples of two of these tumor suppressor genes. In chromosome 17, there is something called BRCA1. And then in chromosome 13, there is another gene called BRCA2. And these are tumor suppressor genes, and these actually suppress growth in certain kinds of tissue, specifically breast tissue. And when these genes are faulty, so if somebody has abnormally functioning BRCA1 and or BRCA2 genes, this substantially increases the risk for cancer and can make the cancer much more difficult to treat. And so somebody who has breast cancer, oftentimes what we'll do is we will look at their genetic profile and look at the BRCA1 and BRCA2 genes and see if they are normal or abnormal. And if they're abnormal, that may complicate the treatment. Or somebody who has a history of breast cancer in their family may preemptively get tested, and they may find, oh, I have abnormal BRCA1 and BRCA2 genes, and they may opt to have like a double mastectomy, for example, have both their breasts removed prior to developing any problems, essentially attempting to prevent, right, or prophylactically treat the cancer. And BRCA actually stands for breast cancer, right? Sometimes cancer is denoted with a CA. And the study of cancer is known as oncology, right? Hence the term oncogene, right? After the term oncology, the study of cancer. So that's just one example of how genetics and environment, or genetics specifically, there are others, but this is a pretty common one that we run into. And it is very common if somebody gets diagnosed with a certain kind of cancer, we often will take a sample of the tumor and look for specific genetic markers on that tumor and try to develop therapies that target those. There are certain kinds of therapies out there. We'll talk about some of them a little later on today, something called monoclonal antibody therapies for cancers. And if the genetics of a cancer are of a certain type, these certain therapies may in fact be incredibly effective for those. All right. So that's just some additional cancer information I wanted to make sure that I covered. So with that in mind, just as a reminder, the genes are what code for a specific trait, right? Genes code for a specific trait. And then a large collection of genes is contained within something called a chromosome. And the chromosome structure is very different from animal to animal. In human beings, does anyone know how many individual chromosomes does a normal human being have?
46.
46, yeah, so 46. So a normal person will have 46 chromosomes, and that actually is 23 pairs of chromosomes. So you get 23 chromosomes from mom and 23 chromosomes from dad, right? And under normal circumstances, when cells normally divide, they copy all 46 chromosomes, right? A cell in your body ends up dividing into two daughter cells that have full copies of the DNA. That is a process known as mitosis. Now, there are special cells in your body, your sex cells. We call this gametogenesis, or the gametes are your sex cells. So in the biofemale, that would be the ova that are developed, and in the male, that would be the sperm, right? And in that case, there's a very special kind of cell division called meiosis, with an E instead of an I. And that is where you just have one, right? You just have a single set of chromosomes. And then one set from mom, one set from dad come together, and you have two full sets in the offspring. All right, so it's mitosis and meiosis. And the way that this works is the 22 of those 23 pairs are what we call the autosomes. All right, autosomes. And then the last pair, the 23rd pair, so 1 through 22 are the autosomes. The 23rd pair are what we call your sex chromosomes, right? And these determine biological sex. And generally speaking, a biological male is going to have 2 X chromosomes. Biological female is going to have an X and a Y chromosome. And there are different disorders involving the sex chromosomes. Excuse me. And that's kind of what I want to talk about now. I want to talk about just a little bit about genetic disorders. They're incredibly common. We just want to have some understanding of the language used. So there are different ways of identifying genetic disorders. You can just identify the disorder by the chromosomes involved, right? So if a disorder involves chromosomes 1 through 23, we call that an autosomal disorder. All right, an autosomal disorder. And if it involves the 23rd pair, right, abnormal genes on the 23rd pair, that is called a sex-linked disorder. All right. There is some other terminology you need to be aware of. There are the terms dominant and recessive. All right, dominant and recessive. And let me just ask you all the question. Does anyone know what the difference, what is a dominant versus a recessive disorder? What does that mean?
You need to have it from both parents to get it. And then in the other one, you just need it from one.
Which is which?
I believe the recessive, you need it from both.
Awesome. So the way that it works is if you have, let's say you have an autosomal recessive disorder, right? So let's say that this involves chromosome 20, right? And I don't know what chromosome 20 looks like off the top of my head, but let's just say this is chromosome 20. So this is chromosome 20 that you got from your mom. And then this is chromosome 20 that you got from dad. And the way that autosomal disorder works is, let's say that the gene for that specific disorder is right here. All right. A recessive disorder is if you have an abnormal, so let's say that red is abnormal. So that's the abnormal gene. And let's say that you have a normal gene. So you get a copy of that gene on the other side from your dad, but that is normal. All right. If the disorder is recessive, you will not develop the disease. We will say that you will have the trait. You will have the trait. You have an abnormal gene, but you will not develop the disease. However, if that chromosome is abnormal or if that gene is abnormal, or those genes are abnormal in both chromosomes, so it has to be abnormal times 2. So if that trait is present in both chromosomes, then you will develop the disease. So that is what we call a recessive, right? Same thing with a sex-linked, right? It could be recessive. And then dominant is if you just have the gene, right? As long as you have that gene, it doesn't matter if you have a normal gene in the other chromosome. If that gene is present anywhere, you will develop the disease, right? So you only need one copy of the gene or genes, whereas in recessive, you need 2 copies of the genes or genes to develop the disorder. So let me just give you some examples. I believe the textbook has some good tables as well, but let me just throw some examples out at you here. So we talk about autosomal dominant, for example. Autosomal dominant, some examples would be Huntington's disease, for example, as a common one. That is a neurodegenerative disorder where people develop what's called chorea, with a C, I believe it's CHOREA, where they develop these abnormal movements, and they end up, it progresses, it often results in a neurocognitive decline, like a dementia, and it is fatal and not easily treated. There really aren't any curative treatments, at least. So that's Huntington's disease. Marfan's syndrome. Does anyone know what Marfan's syndrome is?
Isn't that an abnormal growth? You just keep growing and the whatever gene that turns it off just doesn't work?
No, there are some diseases where you do have abnormal, like, body growth. But Marfan's syndrome is actually, it belongs to a group of disorders known as connective tissue diseases, where you have abnormal connective tissues. So Marfan's syndrome, Ehlers-Danlos syndrome, or EDS, are two examples. And essentially you have abnormal connective tissue, and in a lot of cases you have connective tissue that doesn't work. And so these patients, you know, they tend to, their joints, they tend to be able to move their joints in ways that they shouldn't normally move, quote unquote double jointed. Unfortunately, there are lots of problems associated with these connective tissue disorders like Marfan's syndrome and EDS, where they might be more injury prone, and they have a lot of vascular disorders. Your blood vessels are more prone. You have abnormal development of your blood vessels. Aortic dissections, aneurysms, and things like that are more common in these people. Yeah, so these are connective tissue. It's a connective tissue disorder. So with Huntington's and Marfan syndrome, if you get the gene, doesn't matter if you have a normal gene in the other chromosome, you get the disease. However, some examples of autosomal recessive disorders would be something like cystic fibrosis. Does anyone know what cystic fibrosis is, or CF?
Is it mucus buildup in the lungs?
Yeah, that's one of the things that happens with cystic fibrosis. It is actually a form of COPD. It is one of the forms of COPD that is genetic. There's another genetic form of COPD that is called alpha-1 antitrypsin deficiency, which is genetic. You have a gene that codes for an abnormal alpha-1 antitrypsin, and that makes your lungs more prone to becoming damaged in developing COPD. But cystic fibrosis is a disorder. It's actually a disorder of chloride channels. So there's a defective genes, and those people actually they have abnormal chloride channels, and so they can't move chloride normally across cell membranes. And what that does is that prevents them from developing normal mucus, right? They can't move chloride ions, and it doesn't allow them to move water into certain areas. And so what happens is they end up making a lot of thick, sticky mucus, and you can't get that out of the lungs. And then that builds up in the lungs. It causes obstruction. It tends to cause lots of infections as well. Most people with cystic fibrosis actually die of infections, but it doesn't stop there, because it also involves mucus production in other areas of the body, such as the pancreas. And these people are often not able to produce and to secrete pancreatic enzymes, and so they tend not to be able to digest normally, right? The amylase, the lipases, and so on. And so they tend to have digestive and malabsorption problems. They have these respiratory problems. The way they sweat is abnormal. In fact, that's actually how we detect it. That's how we test for it initially, is something called the sweat chloride test. And so, yeah, cystic fibrosis actually impacts multiple body systems. And we are actually able to now treat certain types of cystic fibrosis with genetic therapy. And so in some people, there's actually a cure depending on the specific genes involved. Another example of an autosomal recessive disorder might be something like, let me think, sickle cell anemia. Yeah. Sickle cell disease, right? Where you have abnormal hemoglobin, you have abnormally shaped red blood cells. They have like a sickle shape. Those red blood cells don't transport oxygen normally. They tend to clump up, right? Because normally a red blood cell kind of has this disc-like shape, or what we call a biconcave-like shape. So if we look at it from the side, it kind of looks like that. You look at it from the top, it kind of looks like that. And that shape allows them to be very flexible, and they can kind of flex down, and then they can kind of pile in through capillaries. Whereas these sickle cells, kind of look like a crescent moon, are very hard and don't move, and they tend to clump up. And when lots of them clump up, they end up clogging up capillary networks, and that causes ischemia to various organs. And when that happens and that happens in large parts of the body, that actually is what we call a sickle cell crisis, right? It can cause kidney damage, heart damage, lung damage, brain damage, and it tends to be incredibly painful. So that's an example of autosomal recessive. There are also there are actually some sex-linked disorders as well. Oftentimes they tend to involve the X chromosomes. One that you might be familiar with is an X. So these are sex-linked. This is an X-linked recessive disorder. And this would be a form of hemophilia. So this would be more common in the biological male. And hemophilia is not just one disorder, it's actually a group of disorders. But in general, hemophilia literally means love of blood, right? Heme, blood, philia, love. But what it is a disorder of clotting factors. You have inefficient or ineffective clotting factors. Certain types of clotting factors are associated with certain types of hemophilia, right? You got hemophilia A, hemophilia B, and some of them involve factor 7, 7A, and so on. So those are just some examples of genetic disorders, and hopefully you all have a little better understanding of the language that is used behind certain genetic disorders, is what I really wanted to focus on there. So then that brings us into just a quick discussion since we've been talking about genetics and cells working and dividing and all that. Let's just go ahead and quickly review what's going on with cells before we take our first break. So when you all took anatomy and physiology, did you all learn the cell cycle? Was that something that was taught in your anatomy class or your physiology class?
Yeah, you're talking about cell division and the different phases.
Yeah, you all learned that?
Yes.
Yeah.
Okay, good. So I'm just going to briefly review it just to make sure everybody's on the same page. So when we talk about cells and cells dividing, there's something called the cell cycle. And the reason I'm bringing this up is this is relevant for cancer, right? Because this involves cell division, and this is really relevant for the treatment of cancer. And this helps us understand why certain chemotherapeutics like the alkylating agents and so on have so many side effects because these drugs target rapidly dividing cells, right? And so the side effects tend to occur in body systems where you have lots of cell division. So where do we see most of the side effects from a lot of these chemotherapeutic agents?
Is it the hair?
Yeah, that's absolutely, that's one area, right? The hair falls out because the hair follicles are rapidly dividing, right? And you got your little follicle, and then you have the basement membranes, and they're dividing and pushing, right? And the cells are dividing and they're pushing up, and that pushes the hair out. And so those rapidly dividing cells are targeted, they die off. And these chemotherapeutic agents, a lot of them are not specific to like cancer. Some are. We're trying to make them more specific, but a lot of them are not. They're just specific to rapidly dividing cells. So yeah, the hair takes a hit. What other areas of the body takes a hit?
Lymph nodes?
You're in the right direction. But your blood takes a big hit, right? Those stem cells that, right, the myeloid and the lymphoid stem cells we talked about yesterday are constantly dividing and cranking out white blood cells and red blood cells. And so those take a big hit, and it's very common for people with cancer that are being treated. It's not necessarily the cancer that's doing it, it's the treatment for the cancer hits their bone marrow, hits those stem cells, and they can't make red and white blood cells. And so what happens? They become anemic and they become immunocompromised. Their immune system doesn't work because they can't make adequate numbers of white blood cells, right? So they are immunocompromised, they're anemic, their hair falls out. And you also tend to have problems with the gastrointestinal tract, right? Because your mucosa is fairly rapidly dividing. And so like ulcers of the mouth, nausea and vomiting, gastrointestinal upset and distress, very common with people that are being treated for cancer as well. And hopefully that makes sense. It's like, oh yeah, because these are cells in your body that normally are dividing very, very rapidly. And so it makes sense that these fairly non-specific agents would target those areas of the body as well. Okay, so let's just talk about the cell cycle. So I'm just going to start arbitrarily at the new cell. So we have a newly created cell here. And just remember, not all cells go through this cycle. Not all cells divide. Like neurons in your brain, they don't divide. So a lot of this may not be relevant, say a neuron or a myocyte in your heart, but it's going to be much more relevant for, say, the basal lamina that's producing respiratory mucosal cells, something like that, where you have lots of cell division. So I'm going to start at the new cell. All right. And so what's going to happen is the new cell is going to grow, right? It's going to grow. It's going to have some growth and it's going to have some development. All right. And that phase, that initial phase of growth and development is what we call G1, right? G1. This is the first phase of growth and development. All right, G1. And then what happens is at some point, this cell, as it grows and it develops, it's going to signal, hey, we need to look at dividing into a new cell. And so what it has to do is, before it divides, it has to copy its DNA. And so it will go into what we call the S phase, right? Which is the next part, right? And so in that phase, it copies DNA and certain proteins that are needed, right? And that is called the S phase, or what we call synthesis, right? And then following synthesis, following the copying, of its DNA, it will go into a second growth phase. And this is just preparing for cell division. And this is called G2, right? And then G2, growth 2, it just prepares for division. And then it will divide. And the entire phase of cell division is known as mitosis, or meiosis if we're talking about sex cells, right? And then mitosis is broken into several subphases. So the first phase of mitosis, shall we say, is what's called prophase, right? And in prophase, essentially what happens is the chromatin condenses into chromosomes, and the nucleolus, the substructure of the nucleus, disappears, right? And so the chromosomes are kind of unwrapped, right? Unwrapped from the histones, and the chromosomes are now being exposed, if you will. That's prophase, right? And then they're actually like subphases, but we're just going to go with the major ones. And then the next phase is what's known as metaphase. All right. And in metaphase, this is essentially what happens is you have two copies of the chromosomes, and the chromosomes get pulled apart, and there are little pieces of cytoskeleton that attach at each end. And so you get something that looks like this. It's really cool if you see it. You look at it under a microscope, you've got your cell that's in the act of dividing. And then you have these little structures called centrioles that move to the sides of the cell. And then you have little pieces of cytoskeleton that actually go out from those centrioles and attach to the chromosomes like this. And the chromosomes kind of make a line right here in the middle, right, of the phase. It's kind of like an imaginary plate, if you will. And that's what happens in metaphase. The chromosomes are pulled apart, and then they're lined up, right? And then the next phase is what we call anaphase. All right. And in anaphase, what happens is these chromosomes get pulled toward this part of this new cell in that direction, and the chromosomes over here get pulled down toward that centriole. And so the chromosomes, they're lined up in metaphase, and then they're pulled apart in anaphase. All right. And then you have what's known as telophase. All right. And in telophase, what happens is the nuclear membrane, it had already dissolved. The nuclear membrane starts forming again, right? So that's in telophase. And you start getting an invagination that occurs. So I'll draw that here. So it kind of looks like this. You get an invagination start happening here. And then the nucleus starts to reform on both sides. And of course, you've got those chromosomes that are inside of the nucleus. All right, that's telophase. And then the final phase is what's known as cytokinesis. Right. And essentially in cytokinesis, it's a little cleft, it's a little invagination that's formed, goes all the way through, and it splits. And now you have two exact copies to daughter cells, right? And that's that. Now, some cells, after they're produced, do not go into this process. They go into what's just called G0, right? Or some people refer to this as the resting phase, right? Or some cells will undergo something called senescence. And senescence is actually associated with aging, and where the cells just kind of shut down and they don't divide anymore. And they'll just kind of go into more dormant state. You see increased senescence as people get older. And there are other things that happen as well. When you have DNA that gets copied, you kind of have these markers at the end of the DNA called telomeres. And every time the DNA gets copied, the telomeres kind of get shorter and shorter. And that can potentially impact replication. You have an accumulation of mutations that can occur, and some mutations can be corrected and some not. But just over time, right, you have an accumulation of mutations, your telomeres shorten, more cells are going into rest phase and senescence. And then over time, right, you have increased reactive oxygen species that are being developed normally. And just over time, that essentially makes these cells less and less likely to divide rapidly, to divide properly, or to copy the DNA appropriately because of accumulated mutations. And so you can see that a lot of this plays a big part in what's known as aging, right? The aging process, as all of these errors kind of add up, if you will. And that increases the risk for cancer. It prevents us from repairing as effectively when we're younger, and so on and so forth. And we start really seeing these changes in the mid-30s is where we start seeing a lot of these significant changes of this kind of this gradual shutdown of senescence errors and things like that. About 35, like you'll start seeing your kidneys right after you lose about 1% of normal kidney function every year after 35. And we normally say that your normal GFR, your glomerular filtration rate is like 80 to 100. And the reason there's that big range is, a 20-year-old, is probably going to be closer to 100. And then maybe you're down in the 80s in your mid 30s. And then by the time you're 60 or 70, it may be down to 60, right? And if you had a GFR of 60 when you're 20, that would be a critical problem. But, if you have a GFR of 60 when you're 80, that may be normal for you, right? And so you see a lot of normal age-related degeneration of body systems, and that is a big part of why it can be so difficult to recover and to manage older people that have injury and illness, not to mention what are called comorbidities. And these are co-occurring diseases like diabetes and heart disease that can further complicate the situation. Okay, so with that in mind, we are coming up on our first break. Are there any questions? Does anybody have any questions?
No, sir.
No, sir.
All right. Well, I think what we'll do is we'll cut it off here then, and I will see you all at 9:45. See you all at 9:45. All right, welcome back. Hopefully everyone had a decent little break. So before we move on, any questions? All right. Send them as PDF format. Perfect. I'll take a look at them here as soon as I can. Appreciate the resending the file. All right. Well, let's move on to the core content I wanted to get through today, and that is talking about immunopathology. So before we dive into some of the major types of pathology, we need to have an understanding of the immune system and how it works. And it's utterly amazing. It's actually just astonishing. We're not going to be able to do it any sort of justice today, but hopefully you all will come off having a much better appreciation for what goes on and some of the things that can go wrong. So let me go ahead and just see about sharing this with you all. Okay, there we go. All right, so let's talk about the immune system. Sure you all can see that okay. All right, looks like you can. All right, so let's talk about the immune system. So the immune system, what are the roles of the immune system, first of all? Well, has several roles, but some of the roles are protect the body from foreign invasion. Pretty straightforward, right? To protect us from pathogens, some of the pathogens we talked about yesterday. It's also to enhance repair of injury or illnesses So whenever you get injured or whenever you have an illness, the immune system also enhances repair, right? That's actually like inflammation we talked about a little bit before. Inflammation in some contexts can help with repair, right? If you twist your ankle, for example, and you injure tissue there, well, what happens? Well, chemicals are released that cause pain. Chemicals are released that cause increased capillary permeability, so nutrients can leak out of the vasculature and into those damaged tissues, and you get swelling, and that swelling and pain helps to immobilize that injured ankle, for example, right? And so the immune system is helping to facilitate that as well. And the third major role or function of the immune system is to identify and deal with faulty cells, right? Cells that aren't working well, or cells that are maybe dividing in ways that they shouldn't be, right? So that is also a very critical part of the immune system. And the coolest thing, or one of the coolest things about the immune system is, aside from the nervous system, this is the only other area where the concept of memory comes into play, right? Memory is an important component of, well, a branch of the immune system at least. And we will talk about that here in just a little bit. So there are specialized immune system tissues and organs, and I just want to cover them a little bit. You have what are called the progenitor cells. We talked about those yesterday. These are the stem cells that then differentiate into the white blood cells. The myeloid, you have the myeloid line, which differentiate into like neutrophils and monocytes. And then you have the lymphoid line, which are your killer T cells, your helper T cells, your B cells, and that differentiate into plasma cells. All right. And just to review types of white blood cells, we talked about these yesterday during hematology. You've got never let monkeys eat bananas. You got your neutrophils, your lymphocytes, your monocytes, your eosinophils, and your basophils. And there are two broad categories that you can divide these 5 types of white blood cells up into, and those include the granulocytes and the agranulocytes. And the granulocytes, when you look at them under a microscope, they look like they have little granules of stuff inside of them, and those are little, basically little bags of enzymes that those cells, enzymes and other chemicals that those cells can release to help modulate the immune response, might help them digest pathogens or enhance inflammation. So the neutrophils, the eosinophils, and the basophils are all granulocytes, right? You can release those little vacuoles of granules. All right. This is some terminology. A lot of this we've already covered. But remember, leukocytosis is an elevation white blood count. Remember, 5 to 10,000 is what we tend to go for normal. Leukopenia, however, is decreased white blood count. Remember the concept of a left shift, right? This refers to the neutrophils. If your white count, if you have leukocytosis and you look at the differentials and the neutrophils are elevated, right? We looked at the banded versus the segmented neutrophils, and that has to do with the nucleus. And we know that the bands are immature, and these tend to increase when there's an acute infection. And so when you see that, we call that a left shift. However, segmented neutrophils are mature neutrophils, right? And we get this off of the manual differential or the differential, and we look at all the individual white blood cells as opposed to just the total white count. So the white blood count, the WBC, right, is just a total count. And then the differential looks at all of the individual never let monkeys eat bananas, all the individual white blood count, white cells. And if the neutrophils are elevated, that's when we look at the banded versus segmented neutrophils. All right. So nothing new there. You also have what are called your lymphatic tissues, right? These are an important part of the immune system as well. So what you have lymphatic vessels. These are little vessels that absorb fluid, right? Pull fluid out of the interstitial space. And then that fluid goes into these vessels. And then these vessels go into large junctions. And in those large junctions, you have little sac-like structures called nodes or lymph nodes. And each of us has approximately 600 lymph nodes throughout our body, and they tend to be concentrated in certain areas, such as the armpit. You have several in the axilla, or we call your axillary nodes. You have several in your neck, your cervical lymph nodes. You have some in your chest, in your lungs, your thoracic lymph nodes, in your groin as well. And you have about 600 or so spread throughout your body. And what happens is that lymphatic fluid goes into those lymph nodes. And within those lymph nodes, you have large concentrations of white blood cells. And those white blood cells essentially kind of filter that lymphatic fluid out, if you will. If there are pathogens in there, they filter it out, they identify antigens, and so on and so forth. And that's why sometimes if you have an infection downstream from a lymph node, you can get swelling of that lymph node because there's that downstream infection. And when you get a swelling of a lymph node, that is referred to as lymphadenopathy. And typically when you have swelling of a lymph node, that means you have an infection. It could be bacterial or viral or parasitic or fungal. You either have an infection, or in some cases, certain kinds of cancers can cause lymphadenopathy, specifically cancers that involve the lymphatic system, and those are called lymphomas, or of the white blood cells, and those are called leukemias. Those types of cancers commonly cause lymphadenopathy. The spleen is also an important organ. It's actually kind of a large lymph node. The spleen function is as kind of a large lymph node in the left upper quadrant of the abdominal cavity. All right, good. You also have lots of tissue in your gut, and your gut is just a word that means all your intestines, your small intestine, your large intestine. And you have a bunch of tissue there. It's called the GALT tissue, and that stands for gut-associated lymphatic tissues. And this is a lymphoid-like tissue. And as you might imagine, you need lots of that in your gut because a lot of stuff from the outer environment, right, and the external environment is going into your body. And so you need to be able to mount a robust immune response because lots of potential pathogens are coming in that way. So that's just kind of the 30,000-foot view of the immune system and what it does. It's a wide, diverse collection of cells and tissues all working together to hopefully do these three major things. All right, well, let's move on. So let's take a more detailed look at the immune system. And the immune system really has two major components to it, all right? And these are what we call the innate immune system and the adaptive immune system. And the innate immune system is also referred to as the non-specific response, whereas the adaptive is referred to as the specific response. So the way that this works is the non-specific or the innate immune system does not have memory. It can't really differentiate. And really what it does is it's good at determining, hey, something's going on here, and I need to start calling the alarm, and I need to start taking the initial steps to protect, right? But it's not very specific. It doesn't have memory. And everybody has a non-specific immune system. Even when you're born, you have a non-specific immune system. And then what happens is, as you come into contact with pathogens, you develop a memory of those encounters, and you develop a catalog as you grow older of, oh yeah, I've encountered this before. And that is really what goes on with the adaptive or the specific immune system. So let's talk about the innate or non-specific immune system. And it has three major components to it. The first major components are the physiological barriers. These are barriers that prevent pathogens from getting into your body, like mucous membranes of your gut and your lungs, and your genital urinary tract, oils secreted on the skin, intact epithelium. Intact skin is a great barrier, right? Tears and stomach acid, right? So these are all things, these are barriers, right? These are the borders that protects, right, stuff from even getting in. And then what happens is if these barriers are breached or they fail and pathogens get in, your front line defense are your phagocytic cells or your phagocytes. And these include dendritic cells, macrophages, and your neutrophils. All right. And these are specific types of white blood cells that we call leukocytes, with the exception of the dendritic cells. The dendritic cells essentially are always in those tissues, right? Just kind of always hanging out in the tissues, whereas the macrophage and the neutrophils are very mobile and they can move throughout your body. And if there's something going on in my hand, macrophages and neutrophils from another part of my body can move into that area, whereas the dendritic cells are kind of stationary, if you will. All right. And these phagocytic cells do what? Well, they phagocytize, right? So they dump out lots of digestive enzymes onto the pathogens, try to neutralize the pathogens, and then they phagocytize those pathogens. All right. And then they do something very special that I'll talk about here in just a minute. All right. So those are the phagocytes. And then the other major part of that is what's known as the inflammatory response. This is inflammation. What is inflammation? Inflammation comes down to a few different things. First of all, you have pain with inflammation. That lets you know something's going on. There's a problem. There is swelling. So you have increased capillary permeability. Typically, that's due to histamine release from mast cells. So you have mast cells embedded in your tissues. And when they sense that there's a problem, an injury or pathogens or damaged or dying cells, they will release histamine. And that causes your capillaries to dilate and become permeable. So white blood cells in your blood can leak out, go out of those and into that tissue that's damaged. All right. So that's part of the swelling. You have an increase in temperature, right? So other chemicals such as cytokines are created actually from the mast cell walls can be after the mast cells degranulate, they essentially rupture. There's there's a component, arachidonic acid within the cell wall that gets metabolized into cytokines and leukotrienes that further regulate or cause inflammation. So you get pain, you get swelling, you have an increase in temperature. This causes erythema or redness. All right, tends to feel warm. And then that also, if an extremity is involved, that helps immobilize and prevent further injury. All right. Now, alongside of that, there is something called the complement pathway. And the complement pathway is a bunch of proteins in your blood. And what these proteins do is when these proteins are activated, they enhance inflammation. And we'll talk about inflammation and complement in a little more detail here in a bit. And so you have various chemical messengers like interleukins enter between leukin and white blood cell, right? Interleukins. And this is called chemotaxis, communication between different cells. You can get something called a MAC or a membrane attack protein that is developed, and it can actually harpoon pathogens and put a little pore in them, and then they start kind of spilling their guts out. Some of these complement proteins will opsonize antigens. And basically what opsonization is they attach to the antigen. and then that acts like a little beacon and allows the phagocytic cells to better phagocytize the antigen. And remember, an antigen is just is a part of a pathogen, right? So you've got your pathogen here, say it's a bacteria, and then there's some protein on the surface of that bacteria that your body can use to identify that specific bacteria, and that protein is called an antigen, right? And so what happens is the complement proteins can attach to opsonize that antigen, and then when your phagocytes come in, that allows your phagocytic cells to better identify and phagocytize that antigen, and then those phagocytes take that antigen into themselves. And then what they do is they go, we can use this to identify this specific bacteria or virus or parasite. And then they take that antigen and they present it over to the specific immune system and say, hey, you can use this to identify this organism and to create antibodies against it. And so phagocytic cells are also known as APCs, or antigen presenting cells. All right. Specifically your macrophages and your neutrophils. And your neutrophils are kind of like your primary APCs, right? And so what they will do is they will present the antigens over to the lymphocytes of the adaptive immune system. So let's just move on over and talk about the adaptive or specific immune system. And this consists of the lymphocytes. These are the primary cells of the specific immune system. And you have two major categories. You have what are called the B lymphocytes. And we call them B lymphocytes because they mature in the bone marrow. And these are responsible for something referred to as the humoral response. And humoral just comes from a Greek word, the humors. And humors refer to body fluids like bile and blood. And so that's where this occurs. This humoral response primarily occurs in the fluids of the body, specifically the blood. And so this occurs outside of the cells. So this is an extracellular response. So what the B lymphocytes do is when the antigen presenting cells present the antigens to them, the B lymphocytes look at them and they go, oh, I am going to make antibodies out of these antigens. All right. And so that's what they do. These B cells become what are known as effector or plasma cells. We mentioned that yesterday after the antibodies presented to them. And then they start producing antibodies that are specific to that pathogen. And then once that infection resolves, the B cells go into a dormant phase. So they quit producing antibodies and they turn into what are called memory cells. And then those memory cells just kind of float around your body dormant. And they can do this for years and decades in some cases. And then if you happen to come into contact with that pathogen again, those memory cells can then spool themselves up and be converted back into plasma or effector cells and start pumping out antibodies again. Now, there's some complex interplay that involves the other part of the specific immune system, the T lymphocytes. So you got B lymphocytes and T lymphocytes, and the T lymphocytes mature in response to hormones from the thymus gland. That's a little gland near the heart. All right, and that's why we call them T lymphocytes. Now, the T lymphocytes are what are known as the cell-mediated or the intracellular response. So this typically occurs within infected cells. So the T lymphocytes are very important when it comes to virus infected cells and when it comes to abnormal cells, such as cells that are maybe precancerous or becoming cancerous, right? It's really important. And the other thing that the T lymphocytes do is the T lymphocytes help activate plasma cells. And so there are two major categories of these T lymphocytes. There are what we call the CD4 positive T cells. So they have the CD4 protein. And what that does is, remember when I talked about major histocompatibility complex, or MHC? This is a major protein system that the immune system uses to identify self versus non-self cells. Now, the CD4 positive T cells attach to MHC2, or major histocompatibility complex 2 proteins. And these proteins are primarily located on these memory cells. And when the CD4 cell attaches, it helps activate them. And so these are also referred to as the helper T cells because of the way they work, their basic mechanism. Now, you also have what are called the CD8 positive T cells. So these have a different protein, CD8 protein. And these attach to MHC1. And major histocompatibility complex 1 is located on other cells throughout your body. And when the CD8 T cell attaches to major histocompatibility complex 1, that signals apoptosis in that cell, right? That signals cell death. And so these are referred to as the cytotoxic T cells, or the NKT, the natural killer T cells. All right. So these cells here, these CD8 cells, are the ones that can identify virus infected or abnormal cells and kill them off before they become a problem. And then the CD4, the helper T cells, help with the activation of plasma cells. All right. And so this is kind of, we've zoomed in a little bit, but this is kind of the big picture of some of the specific processes that are going on. And they're all kind of intertwined and interacting with one another. And I'll just mention a specific disease. It is actually a viral infection, is a virus that actually infects the CD4 T cells and then causes them to go away as they die off. And does anyone happen to know what virus does that?
HIV.
Yep, the human immunodeficiency virus infects the helper T cells. And then as the helper T cells die off, that prevents the appropriate activation of plasma cells. And so that essentially shuts down or severely limits the specific immune response, right? And then over time, as you lose these CD4 cells and as your CD4 level gets very low and you become immunocompromised, we say that you develop something called AIDS, acquired immune deficiency syndrome. And people with AIDS, right, are very prone to infections. So they are immunocompromised and they tend to develop very weird infections that you don't normally see. These are called opportunistic infections. We don't normally see them in healthy people. So like weird fungal infections, and people are more prone to developing cancer. There are certain cancers that are more likely, such as Burkitt's lymphoma, I believe, is one that's common, and Kaposi's sarcoma. Those are two cancers that are much more common in people with age because of their immunocompromise and because these helper T cells are not able to effectively activate the plasma cells. And so you have a reduction in antibody production. All right. Any questions over the big picture there? All right, well, we're going to move on then. So now that we've talked about antibodies, right, I mentioned them, well, let's spend some time going into some specifics, like what are they and how do they work? So let's talk about antibodies. So antibodies. Antibodies are also known as immunoglobulins or Igs. So immunoglobulin is just another term. It is a synonym for antibody. So what do antibodies do? Well, antibodies attach to antigens. They attach to the antigen of a pathogen like a bacteria, a cell, or a virus. So antibodies attach to the antigen on the pathogen. So that, right, so the innate immune system identifies a problem and sends that antigen over to the adaptive immune system. And the adaptive immune system looks at that antigen and creates an antibody based on that antigen, right? And then starts pumping those antibodies out, and those antibodies then attach to the antigen of those pathogens. And so what happens when the antibody attaches? Well, several things can happen. First thing that can happen is the antigen, well, the main thing that happens is the antigen and the antibody. So the antibody binds to the antigen, and that forms something called an antigen-antibody complex. And when that complex is formed, there are four major things that can happen. The first thing is that can activate the complement pathway. And remember, complement pathway helps increase inflammation. It can form membrane attack complex and so on. All right. The other thing that can happen is something called agglutination. So essentially what happens is several of those pathogens can get pulled together. Right? They can clump together and they can agglutinate. And so if you have some pathogens here, and then you've got an antibody that comes in, right? So antibodies attach to the antigens on those pathogens. And then what happens is those antibodies bring all that together. So all that gets brought together. And so you have this big old agglutinated clump of stuff now, and that clump is much easier. It's much easier for the body to identify and get rid of. All right. This also causes antigens to precipitate out of their medium. So they can't stay suspended in the blood if they're in the blood or the tissues, right? So they get agglutinated and they precipitate out, and that just makes it easier to deal with them, right? They're easier to find, easier to get white blood cells to them, and so on. And in some cases, the antibody will just outright neutralize the pathogen. And not all antibodies do this, but some do. And if an antibody is capable of neutralizing or destroying the pathogen, this is referred to as a neutralizing antibody. And it doesn't necessarily have to be a pathogen. It could be a toxin, a poison, or even a venom. And that'll be important when we talk about antibody therapies here in a little bit. All right. So again, over here with the innate immune response, you've got this pathogen here. And this pathogen has some antigen, some fingerprint that the body can use to identify it. So you've got your phagocyte here. Maybe this is a neutrophil. It phagocytizes that pathogen. And then the antigens on the pathogen surface are presented. So here is that phagocyte. It presents those to the B cells. And then the B cells take that and create antibodies based on that antigen. And then those antibodies can specifically bind to those specific pathogens, right? And then the B cell becomes a plasma cell or an effector cell and starts pumping out all these antibodies. And then once the infection is dealt with, then the plasma cell can shut off, quit releasing antibodies, and become a memory cell. But the memory cell can be reactivated at a later time and start pumping out antibodies again if needed, right? So that's where the memory comes in, and that's the concept of immunity or long-term immunity if you are immune to something. So a lot of people think that immunity means that you cannot get infected. It's actually not the case. You can still get infected, but what happens is once you get infected, if your immune system is working, those dormant memory cells can very quickly be converted back into plasma cells, and you can start dumping antibodies out again, and you can rapidly deal with that infection before it becomes a problem, sometimes before people even develop symptoms of infection. All right. So that's what we see there. So let's talk about the antibody structure and function in a little more detail. Go. All right. So antibodies kind of have a Y-shaped appearance to them. All right. And we'll talk about some of these general areas. So you have what are called the heavy and light chains. So you see in blue here, this is the heavy chain. And then it is attached to these lighter chains on the outside of the Y here. And then what you have is, because these are immunoglobulins, these are proteins, right? And proteins are amino acids, right? And you have the nitrogen part of amino acid binding the carbon part, the carboxyl part of one. And so the chain, you have a nitrogen on one end and a carbon on the other end. And so these are the nitrogens on one end over here and the carbons on the other end over here. And we call that the C terminus, the C end, and the N terminus, the N end. And right here, this area right here, let me circle that, is where the antibody binds to the actual antigen, right? And so this is referred to as, this area of the antibody right here is referred to as the Fab domain. And Fab just stands for fragment antigen binding. All right, so this is where the antibody actually binds to the antigen. And this can vary. This is variable. So the the shape of it can vary a little bit to accommodate variations in antigens, right? And there's actually something that happens when you get infected, and as your body's making antibodies, there's something called somatic hypermutation that occurs, where you start cranking out several types of antibodies. And then the antibody that is the most effective for whatever the pathogen is becomes the dominant antibody, right? And so your body makes several different types of antibodies, and some are more or less effective. And through trial and error, the body figures out, well, what is the best antibody for this specific antigen? And so you do have some variability there. And then this here, this region down here is what we call the FC domain, or the fragment crystallization. And this doesn't really change. All right. So that's kind of the basic structure. And then you have all of these intermolecular forces that are holding it together. For example, right in here, you've got covalent disulfide bonds and various non-covalent intramolecular forces like hydrogen bonds and van der Waals forces and induced dipoles and so on and so forth. The reason I bring that up is because what we can do is we can actually break the antibodies up. And so you can separate the FC domain from the Fab domain. right? So if you have an antibody here, and this is the Fab domain up here and the FC domain here, we can chemically separate that, separate the FC from the Fab, and then we can concentrate the Fab and we can make therapeutics from that. For example, antivenoms. One of the most commonly used antivenoms, at least here in the United States or North America, is something called Crofab. Crofab. And that stands for crotolid fragment antigen binding. Crotolids are a type of snake. They are pit vipers. Pit vipers are crotolids. And so what you do is you take venom from several different types of crotolids, and then what we do is we inject that venom into animals. So we take several different types of venom, we inject it into animals, and then animals create antibodies or immunoglobulins. And then we expose those antibodies, we pull those antibodies out of the blood plasma, and we expose them to chemicals that break the antibodies up, and we concentrate just the Fab domain of those antibodies, because that's the most active part. And then we package that in a vial, and that becomes the antivenom. That's how most of the antivenoms for things are created. We can also we can do the same thing for different drugs to treat drug toxicities. There's a drug called digoxin or lanoxin. It belongs to a class of drugs known as the cardiac glycosides. And there is a drug called Digibind, which is an antibody that binds to the drug digoxin and deactivates it. There are also what are known as monoclonal antibodies, and these are essentially a very specific type of antibody that binds to a very specific site. So there's not the variability that you'd see in certain cases. These are very specific antibodies, and we can manufacture them to treat specific problems, certain kinds of cancer. There's a drug called Rituxan or rituximab, for example, which is used to treat a very specific kind of cancer. It is a kind of non-Hodgkin's lymphoma. Lymphomas are lymphatic cancers. And a certain kind of lymphatic cancer, there are two major categories of lymphomas. There's Hodgkin's lymphoma. We sometimes call that Hodgkin's disease. And then there's non-Hodgkin's lymphoma. And then there are subtypes, like under non-Hodgkin's, you've got Burkitt's lymphoma and others. But there's a specific genetic type that Rituxan is very effective at treating. In addition to this, we can create monoclonal antibodies for different viruses. And in fact, that is one of the treatments for COVID-19, right? For COVID-19, right? The virus that causes COVID-19 is the SARS-CoV-2, SARS coronavirus 2. And we can actually manufacture these monoclonal antibodies that are very specific to that coronavirus. Unfortunately, the coronavirus, as it is created within virus-infected cells, it mutates. And so you get what are called variants, right? And the current variant, the dominant variant, or the so-called Omicron variant, is genetically and it's different enough. Its shape is different enough. There's a specific protein on the coronavirus called the spike protein, which is what most of these antibodies, it is the antigen that the antibodies combine to. The shape of that antigen is sufficiently different enough that most of the current monoclonal antibody therapies that we have are not very effective. And so viruses through mutation can also develop resistance to different therapies. as well, in a somewhat similar to what we see with bacteria. But I wanted to bring that up, right? That there's a lot of therapeutic potential around antibodies. So let's talk about the general classes of antibodies. I'm sure you all have probably encountered these before, but I just want to review them before we take our next break. All right, so you have these broad categories of antibodies. So the first is what we call IgG, immunoglobulin G. And this is the most abundant antibody in the body, okay? And this is the antibody that crosses the placental barrier. So mom can actually give some of these to the baby, and this can provide some protection to a newborn, because when you're born, you do not have an adaptive immune system, because your immune system has never encountered any infections. And so these antibodies kind of create a temporary protection while that baby starts spooling up their immune system, right? And so this is very important in the first 6 to 12 months of life. IgG antibodies are very good at attacking extracellular viruses, toxins, and poisons, and as well as activating the complement pathway. All right, that's IgG. IgM are the largest antibodies. And in fact, IgM antibodies are 5 Y-shaped antibodies joined together. So an IgM looks kind of like this: 1, 2, 3, 4, 5. That's what an IgM antibody looks like. And IgM antibodies are released early in an infection. So remember when I talked about that somatic hypermutation that happens, where the body tries to find the best antibody for the pathogen? Well, that takes time. That can take days, sometimes weeks. I mean, if you have an acute infection, it's not like you have that much time. And so what the body does is it starts cranking out these antibodies very quickly that they don't have a high binding affinity to the pathogen, but they're kind of like a stopgap. And that's what IgM antibodies are. They're kind of stopgap, they're large, and they really help with agglutination during the early phases of an infection. And so early on in an infection, your IgM antibodies increase first, and then they kind of agglutinate and pull pathogens together, and that allows the body to develop a more specific response. And then what happens is you start pumping out typically IgG antibodies that are much more specific and much more neutralizing. And so you get a more mature response. IgA antibodies, however, tend to be found on mucosal surfaces. So think of your saliva, tears, blood, your respiratory tract as well. These are also found in breast milk. So mom can give some of these to baby. And these are important when it comes to respiratory infections. In fact, this may be one of the reasons why the COVID vaccines, right? When we talk about COVID vaccines are not particularly good, at least with the current variants, are not particularly good at preventing symptomatic infection, right? So you can still get infected and you can still develop symptoms, right? You cough, runny nose, maybe a fever. And one of the reasons that may be is that the vaccine is very good at causing lots of IgG antibodies, but these are in your blood, right? But not necessarily lots of IgA antibodies. So what happens is you get infected because the IgA antibodies, you don't have as much of that protecting your respiratory mucosa, but that infection can't get into your bloodstream and cause problems throughout the body because you have the IgG antibodies. And so even though the vaccines may not prevent symptomatic infection, they are very good at preventing severe infection, right? So yeah, you're going to get infected and you're not going to feel very well, stuffy nose and all that, but you're not going to get critically ill. You're not going to get ARDS. You're not going to be in the hospital. And this may be part of it. So you can see that there's a lot of complicated stuff going on here. IgE antibodies, the next major categories, and these are also found in the lungs and in mucous membranes in the skin. And these are very good at reacting to foreign substances, and it is the IgE antibodies that are primarily responsible for allergic and anaphylactic reactions, right? So allergies and anaphylactic shock. And then the last major category of antibodies are the IgD antibodies, and we don't understand those very well. We're not quite sure what they do, what they're involved in. Okay, so before we take our next break, are there any questions about anything we've gone over so far? Okay, well, let's see. It's 10:32. I will see you all at 10:45. Okay, we'll go ahead and get going again. And this section of the lecture will be fairly short, I think. A couple more things I want to talk about, and then we'll wrap it up for the day. So I just wanted to spend a few minutes talking about the complement pathway. And we're just going to take a very surface view of complement. But just remember that it is a collection of proteins. And when these proteins are activated, they can do a few different things. And we generally say that there are three major pathways by which this activation can occur. You've got something called the classical pathway. It was just the first pathway that was discovered. That's why we call it classical, just historical. And then you have what's known as the alternative pathway. And then there's yet another pathway called the mannose binding lectin pathway, or the MBL pathway. So there are three ways that it happens, but essentially what you get is you get the following things, right? You get opsonization, right? So over in the classical pathway, this gets activated when antibodies bind to the surface of antigens. So when you have an antibody antigen complex that forms, you've got the C1, that's the complement 1 subtype protein, binds to the antibody, and that forms an enzyme called C3 convertase, and that forms a C4B2A complex. And essentially what that does is that allows for the activation of C3 here. And when C3 gets activated, that helps enhance the inflammatory response. And that stimulates mast cells to degranulate. They release more histamine. The capillaries become more permeable, and white blood cells are able to better get out and go into the area of that infection. And it's really cool. Like the neutrophils can literally kind of attach to the surface. So if this is a blood vessel, and then you have an area of the blood vessel that is opened up and permeable, the neutrophils can literally attach to the surface, to the inner surface of the blood vessel. And then what will happen is they'll just kind of like roll along the inner surface. They'll roll along until they get to the little opening there, and then pop on out and go into the tissues. And that collection, or that infiltration, and as they roll, that's called diapedesis. And you can actually look at this. You can take a slide of this if someone who's maybe died of an infection or something like that, and you can see this histologically occurring. It's really kind of interesting. Okay, and then the alternative pathway, it's similar to the classical pathway, but it just involves some other proteins like MASP, MBL, mannose binding lectins involved there. And essentially what happens is these proteins bind to sugar molecules on the pathogen. So the pathogen can have glycosylated proteins and things like that. Glycosylated just means there's a sugar attached to it. And that's mannose is actually a type of sugar. It's an oligosaccharide. And that just forms C3 convertase, the same C3 convertase over here. And so C3 can be activated into C3A, where you get inflammation modulation, or you can get C3B, And remember, C3b can either cause opsonization, which the opsonization just means that these complement proteins bind to the antigen, and that just allows the phagocytes to better phagocytize that specific pathogen. And then the other thing that can happen is as you're forming C3b, is you get the MAC, the membrane attack complex, which is a little pore that's formed and just punches a little hole in the pathogen. So, something like a parasite or a bacteria, this is where you would see this become relevant. Viruses, maybe not so much. All right. So anyway, that's just the complement pathway. It complements or helps the immune response, specifically the innate immune response. But as you can see, right, the antibody response can actually help activate complement as well. So there's a lot of crossover going on here. The last thing I want to talk about some different pathologies associated with the immune system. And we've talked about some already, but these are specific, what we call hypersensitivity reactions. So these are essentially the immune system not being able to identify self cells, a self versus non-self problem. And these are referred to as the hypersensitivity reactions. And there are 5 major types of hypersensitivity reactions, and I just want to review them real quick. All right. So the first, and by far one of the most common, is what we call the type 1 hypersensitivity. So if somebody has a type 1 hypersensitivity reaction, this is mediated by IgE, right? Immunoglobulin E antibodies. And this is responsible for the seasonal allergies and anaphylaxis, right? So the allergies that a lot of us have, the severe anaphylactic reactions, these are IgE mediated. So that's a type 1 hypersensitivity reaction. A type 2 hypersensitivity reaction is something that we call an antibody-mediated cytotoxicity. So what happens here is an antibody attaches to a cell and causes destruction of the cell. So you have direct cellular destruction occurring. So type 1, you do not have cell destruction. In type 1, what you have is you have an over-exaggerated inflammatory response, right? So you have lots of increase in the capillary permeability, right? Fluid leaks out of your blood vessels into the interstitial space, and you have swelling, right? You get angioedema of the airway, right? Your blood pressure gets low, you have distributive shock, but you're not having direct destruction of cells like you are with type 2. So some examples of type 2 hypersensitivity reactions are the ABO, right? The blood transfusion reactions, the hemolytic reaction, where, right, you have with your blood cells have different antigens, and the major antigens are the A antigen, right? The B antigen and the D antigen. The D antigen is also referred to as the Rh factor. So if somebody has type A blood, right, or A positive, and positive just means that they have the D antigen. So A positive would mean that they have A and D antigen on their red blood cells. And that blood comes into contact with somebody that has type B blood, right? The person with type B blood, their immune system is going to recognize the A antigens as being foreign and mount a cytotoxic defense, right? So that's what you have. You have hemolysis. You have destruction of the red blood cells. They break down. We see this in various hemolytic newborn diseases, specifically where a mom who is Rh negative has a baby that's Rh positive. And so the mom's immune system may attack that Rh positive baby. There are some drugs that can attenuate this, like Rogam can be given to help prevent this from occurring. So this is actually an important part of a prenatal workup for a mom is what is mom's RH? Is she RH positive or negative? Does she or does she not have the D antigen? All right. There are also drug-induced hemolytic anemias where a certain drug is given and then that drug binds to proteins on red blood cells, and that drug protein complex, the body, the immune system goes, this looks like a foreign invader. I'm going to mount a response here. And antibodies are produced. And then the body starts attacking the red blood cells. Penicillins and cephalosporin antibiotics can actually cause this to occur. So that's type 2 hypersensitivity, is direct cell destruction. Antibodies are created that attach to cells and destroy them, oftentimes red blood cells. Okay. Now, a type 3 hypersensitivity reaction is something known as an immune complex induced inflammation. So this is not direct cell destruction. This is a problem of too much inflammation. And remember, with inflammation, we have the creation of lots of reactive oxygen species and free radicals. So this induces a lot of oxidative stress in those tissues that are chronically inflamed. And it is this oxidative stress that over a long period of time causes the damage. And there are many examples of type 3 hypersensitivity reactions, lupus being one of them, or this is also called SLE, systemic lupus erythematosus, rheumatoid arthritis, right? Where you have chronic immune complex induced inflammation of the joints, good pastures and post-strep glomerulonephritis. And so this tends to happen in the setting of strep following a streptococcal infection, like strep pneumoniae or something like that. What happens is you can have this immune complex that forms, and it causes chronic inflammation in the kidneys and other areas of the body. All right. So those are some examples of type 3 hypersensitivity reactions. Type 4 hypersensitivity is something referred to as a delayed hypersensitivity. This is something that can take several days to develop, typically 2 to 3 days. And so classically what happens is you come into contact with something and then it gets absorbed into your skin typically. And then what happens is that molecule combines with other molecules in your body. All right. And that new molecule that's formed is identified by the immune system, and then the immune system mounts immune response. This is a type 4 contact dermatitis. So you come into contact with something, and the classic example of this is poison ivy and poison oak exposure. So what happens here is you've got something called a hapten, and a hapten is just a small molecule that by itself doesn't do anything. But what happens is that little molecule gets absorbed, so that there's this molecule called urushiol that is in the poison oak and poison ivy, and that gets absorbed in. All right. And then what happens is that urushiol attaches to large proteins within the skin, and that creates something called a hapten carrier adduct. And that more complex molecule that's created is actually what the body identifies as being problematic. Or sometimes that molecule is inflammatory on its own. And so the urushiol will attach to proteins in your skin and become a highly reactive molecule called quinone. And it is this quinone that causes the manifestations that we see, right? The itching and the rash and the breakdown of the skin. But it takes a few days for that to, get absorbed. The oils of the ivy or the oak get absorbed into your skin. The urushiol attaches to the proteins. The hapten carrier adduct's created, and then the quinone is created, and then you start having manifestations of the disease. And then finally, yes.
Hey, can you explain why some people are unaffected by poison ivy and then others are severely affected? Does it have something to do with these haptens? Or?
Yeah. To some extent, we don't exactly know why, but everybody's genetically a little different, and so the proteins that they have in their skin, the proteins that the urushiol connects to may be a little different. And so when it attaches, it may not be able to attach, or the adduct that's created does not result in the creation of the quinone, that reactive molecule.
Got it.
So, yeah, no problem. A lot of it's genetic, and sometimes sometimes people need repeated exposure. So some people, may not develop a whole lot following exposure, and then maybe the 5th or 6th or 7th time they get exposed, then they end up developing and they manifest the contact dermatitis. All right, good deal. And then finally, the last type of hypersensitivity reaction is what we call a type 5. Type 5 is similar to type 2. So remember, type 2 is where the antibody attaches and causes direct cell destruction. But the big difference with a type 5 is the entire cell is not destroyed. Part of the cell is destroyed. So the difference between a type 2 and a type 5 is a type 2, the entire cell, the entire red blood cell hemolyzes, destroyed. A type 5, one part of the cell is attacked. Oftentimes it is a channel or a receptor that gets attacked. Okay. It's oftentimes a channel or a receptor that gets attacked. So we'll talk about some examples. A common example is something called myasthenia gravis. Myasthenia gravis is a neurological autoimmune disease where antibodies created by us, autoantibodies, attach to acetylcholine receptors and destroy the acetylcholine receptors. So they don't destroy the cells, but they destroy the acetylcholine receptors, which means that the muscle cannot respond to acetylcholine being released. And so that prevents proper muscle contraction. And people with myasthenia gravis who develop myasthenia crises and things like that, right, lose the ability to control their muscles, and they can actually develop a paralysis that starts up top and works its way down the body. And so myasthenia gravis, we all often say, produces a mind to ground myasthenia gravis, or a descending paralysis. And this can be particularly nasty if it involves the airway, right? People lose the gag reflex. They can't protect their airway. They can't control their secretions. Or if it gets into the chest and takes out the intercostal muscles or the diaphragm, they may lose the ability to effectively ventilate as well. But again, cells aren't being destroyed, rather the acetylcholine receptors are being destroyed. Another example would be pernicious anemia, where a specific protein gets attacked, something called the intrinsic factor protein, which prevents us from absorbing vitamin B12, right? And that is one of the ways that you can develop anemia, right, is by having decreased vitamin B12 absorption. We will talk about anemia in much more detail in hematology. Graves' disease, morbus Graves' disease, is a type of hyperthyroidism. And what happens with Graves' disease is antibodies attack the thyroid stimulating hormone binding receptor, the TSH binding receptor, that gets attacked, and that causes an overactivation of the thyroid, which causes the thyroid to release too much thyroid hormone, T3 and T4, and that increases the metabolism of your body, and you get tachycardic, and you lose weight, and you have fat redistribution, behind your eyes, and you get the exophthalmos, or the bug eye appearance, and the dry eyes, and the tachydysrhythmias, and the hypertension, and all of the manifestations that you see with Graves' disease. All right. So those are some examples of the type 5 hypersensitivity reaction. And these are collectively often referred to as autoimmune disorders, where the immune system attacks itself, or attack the body, the body's immune system attacks its own cells. And these are the 5 major categories of these autoimmune disorders. And these are very common, very problematic, hard to treat in many cases. And oftentimes the treatment involves suppressing the immune system. So people that are being treated for autoimmune disorders are often immunocompromised because they're receiving steroids and other immune modifying drugs like methotrexate and others, right? So that's yet another problem that we need to look out for in patients that are being treated for autoimmune disorders. In addition to this, autoimmune reactions may be the basis of other diseases. A classic example is actually IDDM, or insulin dependent diabetes mellitus. One of the major hypotheses behind IDDM is following typically a viral infection in a child. After a child recovers from a viral infection, their body creates a bunch of antibodies, and in some instances, the antibodies, while they're originally being pumped out to attach to the virus or attach to virus-infected cells, has enough affinity to bind to cells within the islets of Langerhans within the pancreas and take out the insulin-producing cells. And so there is a very common history that we see in juvenile or child onset diabetes, where they, within a few weeks of recovering from a viral infection, they're now in the hospital, or you're getting called as an EMS provider to take care of them, and they're in DKA, right? And they don't even know that they have diabetes, or the family doesn't even know. And so there does appear to be an autoimmune component to something like diabetes, at least type 1 diabetes, and certainly other diseases may have an autoimmune component to them as well. In fact, there is a term that we call atopic. An atopic just means a disease or a disorder that has an allergic component to it. Typically, that's going to involve the type 1 hypersensitivity reaction. For example, asthma. Many people with asthma also have a history of allergies and or anaphylaxis. And in fact, it is oftentimes the type 1 hypersensitivity reaction that acts as a trigger for a quote unquote asthma attack. All right. So that's yet another connection that we can make. So you can see there are lots of connections, lots of different kinds of pathology that derive from dysfunction of the immune system. In addition to this, we also see this with infections. Oftentimes when people are sick, the substantial morbidity and mortality associated with certain infections may not be the virus, for example, itself, but it may be the immune system's reaction to the virus. For example, take COVID-19. Many people who die of COVID-19 are not dying in the first week they get infected, right? And those of you that have transported these patients, you see this, right? The first week that you get infected with SARS-CoV-2, you have stuffy nose, fever, chills, right? You have viral stuff going on. And then what happens is typically, as your body starts clearing the virus and you're getting over the viral infection, you have an abnormal immune response and your immune system starts cranking out lots of molecules that cause an over-exaggerated inflammatory response, for example. And then that causes damage. There's so much inflammation, there's so much oxidative stress going on that it damages the lungs and can cause ARDS. And this is sometimes referred to as, let's see, I'm kind of running out of space here. This is sometimes referred to as a cytokine storm. And cytokines are just a class of molecules that the immune system uses to communicate with white blood cells. And these include like interleukins, right? Like IL-6, IL-10, IL-4, there are various different interleukins, prostaglandins, and so on. And so it is actually the immune system's over-exaggerated response that seems to be hurting lots of people that develop COVID. And so we see that most of the morbidity and mortality with COVID occurs 2 to 3 weeks or more after initial onset of signs and symptoms. So there's a lot of stuff that can come back to problems with the immune response. And honestly, it's a miracle that the immune system works as well as it does. So it really should come as no surprise that something that is working all the time is so complicated and doing so much stuff, it should really come as no surprise that there are lots of disorders around this system, because if it doesn't work perfectly all the time, you're going to have some sort of problem or some sort of disorder. All right. So I think with that in mind, that is all that I want to get through today. So before I let you all go, are there any questions? All right. I don't see anything in chat. Very good. So before we go, I just want to remind you all, next Tuesday is going to be our last day of pathophysiology. We will have a lecture on Tuesday, and the final exam will open up after the lecture. The final exam will be in Platinum Planner through EMS testing. Is everybody familiar with EMS testing?
Negative.
No.
I don't know.
No? Okay. So on Tuesday, I will take you all through that component of Platinum Planner. Most of you should be, because is that not what you use during the basic class?
Platinum Planner, yes, but not for any testing.
Oh, you didn't take any tests on Platinum Planner?
No, sir.
No. Okay. Well, it's just a section of Platinum Planner called EMS testing. So you'll log into it and your final exam will be there. It's 100 questions, 100 question final exam, multiple choice. And I believe you'll have about a minute per question. So you'll have about 100 minutes, and it'll be open all day Tuesday. So you can take it. Once you start, you have to finish though. So you do not want to be on shift. You do not want to have a place where your internet connection is not working well, because that could potentially be problematic for you. So once you log in and you start the test, you want to just continue all the way through. So we will talk about that in some more detail on Tuesday after lecture. And let's see, the third chapter summary will be due, I believe, on Tuesday as well. So make sure those are done. There will be no additional homework. And oh yes, And there is going to be a quiz that is going to open in Canvas. So as soon as I'm done with you all here, log into Canvas. Under the quizzes tab, you will see it's going to be a comprehensive quiz. I think it's like 30 or 40 questions. So it's a pretty big one. And you'll have the weekend. So you have the whole weekend to go through that. And that is a comprehensive review, and it's going to give you a good idea of where you stand as far as your understanding and retention of the concepts that we've covered in the class so far. So that will open momentarily. Any questions before I let you all go? Okay.
No, sir.
All right. Well, let's call it, and I will see you all on Tuesday or a Monday, perhaps, if you need to log into my open office hour. All right. Take care, everyone.
Hey, Chris, real quick.
Yes.
So with our previous quizzes, I think we had like 2, I want to say. Could we go back and review them?
The previous quizzes.
Or I think they're like exams. Like the first one, it was like 13 questions, but had like multiple options to select. So I was kind of curious.
Oh,
if we
yeah.
could go.
Why, I did review those.
No, but for like us, so we can go back.
Oh, I see. No, unfortunately not. Yeah, just because the next class is going to potentially get those questions. So those questions.
Oh, okay.
Yeah. Yeah, I see what you're saying. Yeah, unfortunately not, just because those are, yeah. We don't want that to get out, so to speak.
Right. Okay.
Yeah. All right. Any other questions? All right. All right. We'll see you all in a few days.